Hi dear students, this is Miss D.M. Arvekar, working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. We have already discussed about the nucleic acids, the structure of a DNA, structure of a RNA, and here we will discuss about the genetic code. This term it is very special as it indicates that there is certain amount of information which is situated in this term. Before starting the actual lecture, we must clear about this central dogma of our life because whatever processes takes place in our physiology, body physiology or the biochemistry that will happen due to these three steps or which is nothing but the central dogma of life. According to the central dogma, the DNA that is a deoxyribonucleic acid it will maintain its copy by the method of a replication when the cell will get divided. And so the information flow will get transferred from the parental cell to the daughter cell as well as whatever the hormones or whatever the enzymes are synthesized in the body, their nature is a protein. So how that proteins are formed? Because once the proteins are formed, they will get turned off after they will their turnover get over or their lifespan will get over. So these proteins are required in various activities at various duration in the body. So how these proteins are formed? Proteins are different biomolecules and the nucleic acid is the different biomolecules. But the formation of a protein is totally dependent upon the DNA. That is a nucleic acid. So I can refer this DNA as a master molecule. So there is a big question that how these proteins are formed into the cell. These proteins are formed by the utilizing information coming from the gene. Gene is a specific or the segment of a DNA having a particular function. But it is not like that the DNA will be directly converted into the protein or the information from the DNA is directly utilized into the protein. No, never. Because proteins are synthesized in the cytoplasm and the DNA is situated into the nucleus. So there is another question that how this information flow happens from the DNA to the proteins. So in order to transfer the information from the DNA for the synthesis of the proteins, that information is transferred with the help of RNA. So the DNA is utilized for the formation of a RNA. Information on the DNA is converted into the form of a RNA and that mRNA, tRNA and rRNA are moved further into the cytoplasm and that are utilized to synthesize the proteins. And once the proteins are synthesized, the RNAs get degraded. So the formation of a RNA by using DNA as a template, this process is a process of transcription and process of formation or process of synthesis of the proteins by utilizing information coming from the RNA, that is the process of a translation. So overall the central dogma shows three steps which are very important for any cell. One is the replication of a DNA, second is the transcription of a DNA and third is the translation of a RNA. So replication, transcription and translation, these are the three important processes of the life. So there is a big question that how the RNA transfer the information to the synthesis of the protein. So these are the single letter code of the nucleotides and this is the double stranded DNA template, short template. When the transcription will happen, so one of the strand will be used as a template and the complementary bases are added during the transcription. So complementary base of the C is G, complementary A of A nitrogen base is the uracil because this is the RNA. So in this way, the RNAs are synthesized, that may be the tRNA, mRNA, rRNA, and that RNAs are modified and then modified RNAs are transported into the cell cytoplasm to synthesize the protein. Now the proteins are indicated by the single letter code, that is V for valine, H for histidine, L for leucine, T for tar, T for threonine, P for proline, like that. So this is the central dogma, that is the transcription process of a transcription in which RNA is synthesized by using DNA as a template, and proteins are synthesized by using the information from the RNA. So the question arises how it will happen in the cell so that this message in the form of a nucleotide is translated into the message of amino acid. So, like suppose one is not knowing the German language, but he know or he can speak in English, but the people in front of him is the peer is that nature that can only understand the German. So this guy will speak in English. There will be the translator who translate that German into the English, that English into the German, and that German will get communicated to that people. And in this way, the communication will happen. So similarly, in the cell also, how this communication will take place, because the message is in the form of a nucleotides, and the information which has coming into the form of a nucleotides, that to be utilized to form proteins which is different nature, that is having a amino acid. So how it will happen, this was the very big question in the 18th century, 19th century. And so number of discoverers, number of experts, or the number of scientists gave their important time to invest this particular codes to crack this codes. This code is nothing but, or the genetic code. This is the genetic information, and this is read into the triplet of a nucleotide. So this is the triplet. This is another triplet. This is another triplet. So it is always used to translate the message in the form of a triplet. So this triplet of a nucleotide is nothing but the genetic code. So this is a genetic dictionary formulated with the help of different experiments, that may be the triplet binding assay, or the experiment by the Indian scientist Hargobind Khorana and the other well-known scientists. And so from this genetic dictionary, we know that there are different types of codons are there which will code for the different types of amino acids, and in this there are 64 codons. Out of that, these three codons are termination codon, that is they will not codes for any amino acid. Okay, so we will in this lecture we will just discuss about the characteristic feature of a genetic code. The first characteristic feature of the genetic code is a triplet. So whenever the tRNA will bind on the mRNA, that will read the triplet or the trinucleotide set of the trinucleotide set on the mRNA. So this trinucleotide set is called as a codon. So this triplet codon has a 64 codon, and which are enough to code for the 20 different amino acids which will be utilized to synthesize the protein. Now another feature of the codon is it is a non-overlapping. That is, when the mRNA is read by the tRNA, it is never read like that. GUG, then UGC, then GCU. This will be the overlapping, right? So it will not read as in this manner. It will read just GUG, then another next three CAU, then the next three CUG. It will never read that GUG, then again UGC like that. So the genetic code is non-overlapping. So here also, whenever the genetic code is read during the translation, it is in the non-overlapping manner. Another one, whenever you are writing the genetic code, there should be not no any commas will be there, no any colon will be there. So genetic code is a commaless. We will not use any comma whenever we will continuously writing the sequences of the mRNA. We will never use any commas. Okay. So another characteristic feature of a genetic code is it is a universal. That is, whenever you will observe the genetic code, any genetic code in any insect or any another animal belongs to the different classes, that class Pisces or amphibians or the reptiles or the mammals or the plants, algae, fungus. So the genetic code will be always universal, or always same for any um, every organism. But there are certain there are certain exceptions for this. Then the next one is genetic code has a polarity. So whenever you are writing the single genetic code, there should be the 5 prime and the 3 prime poles. 5 prime it indicates that the fifth carbon of this sugar is a free, it is not bound to any other moiety, and this 3 carbon is a free which is not bound to any other moiety. So 3 prime 3 carbon is a free here and the fifth carbon is free here. So this is the 5 prime and 3 prime. It is always read from this 5 prime to 3 prime and never from 3 prime to 5 prime. When you will read from it from 3 prime to 5 prime, the meaning will get changed. This code will become GUC. It will never happen in the molecular biology. It will read from the CUG, that is from the 5 prime to 3 prime always. Again, there are two different terms which are very difficult to remember. One is a non-ambiguous and the second is a degenerate. So, non-ambiguity or the non-ambiguous is the characteristic feature of a genetic code. It means that one for one, that is, there is only one codon which will code only for one amino acid. I will repeat, there is only one codon which will code for only one amino acid. That is, one codon will code for only one amino acid. Not one codon will code for number of amino acid, it will code just for one amino acid. For example, the AAA code will always code for the lysine. That is, whenever the triple A come on the mRNA, only the lysine will be added during protein synthesis. Or the CUC will code for the leucine, like that. That is one for one. Another characteristic feature of a genetic code is a degeneracy or the redundant. It means that there are number of codons for single amino acid. There are number of codons for single amino acid. For example, serine amino acid, it can be coded by codon UCU, UCC, UCA, UCG. Like that, the proline amino acid will be coded by the number of codons, that is CCU, CCG, CCA, CCC, like that. So there are number of codons are coding for a single amino acid, and the single codon will code for the single amino acid. Okay, so degeneracy is that is many for one, many codons for one amino acid. So that's it for today. So in summary, we will summarize this point as the genetic code is nothing but the triplet of a nucleotide which are present on the mRNA and that can be read during the translation to convert that message into the form of a amino acid. And that message is read by the with the help of a tRNA and the ribosomal assembly. We have discussed about the characteristic feature of the genetic code. Genetic code is a triplet. It is always represented in the trinucleotide form. It is non-overlapping. That is, it is always read in the set of three. It is commaless. Whenever we are writing the genetic code, there should be no any comma during the writing. It is a universal, but there are certain exceptional cases. Again, it has a polarity. That is, it is read from the 5 prime to 3 prime. It is non-ambiguous. That is, one codon is only coding for the one amino acid, and it is a degenerate, then it means that there are many codons for coding for the one amino acid. Thank you.